a pipeline for a primate projectome, mapping every individual myelinated axon across the whole brain. Greg Wildenberg, Kevin Boergens, Viktor Nikitin, Alex Deriy, Francesco de Carlo, Vincent de Andrade, Xianghui Xiao, Narayan Kasthuri. DOI https://doi.org/10.10.2023.563679. This article is a preprint and has not been certified by peer review. What does this mean? Abstract. Full text. Info/history. Metrics. Preview PDF. 1 abstract. We developed a pipeline to detail the morphology and complete trajectory of every long-distance projecting neuron in large volumes of the non-human primate brain, a projectome. A projectome would organize how the hundreds of brain regions in a primate brain communicate with each other and provide a comprehensive classification of long-distance neurons. The reason such projectomes do not exist already is that there is no single imaging technique with the resolution and sampling depth capable of producing one in primates. Here we address that gap by combining new ways to cut brains with minimal tissue loss, nanoscale X-ray microscopy imaging that reveals every individual myelinated axon, MA, and the location, size, and morphology of every neuron, and algorithms for tracing individual myelinated axons for millimeters to centimeters across tissue gaps. 2. Introduction. A fundamental goal of neuroscience is to determine the organizational logic of brains. Brains are obviously parcellated, and in some large brains like the non-human primate, i.e. rhesus macaque, and human, there are likely hundreds of anatomically and functionally distinct brains regions. 1. 2. Decades of experiments have suggested that information moves across these brain regions in specific ways critical for brain functioning. 3. And disruptions of these pathways results in a range of neurological disorders, e.g., multiple sclerosis, autism, epilepsy, schizophrenia, aphasia. 4, 5, 6, 7. However, our understanding of how brain regions communicate is biased, limited by existing technology. For example, we have good models of the average trajectory of thousands of long-distance myelinated axons, MA, imaged with MRI, 7, 8, or of the long-distance targets of a small fraction, 1%, of individual neurons, revealed with optical microscopy 9, 10, 11, 12, 13. Such data is clearly valuable, for example see Van Essen diagrams. 3. But these techniques leave open questions about brain-wide organizational logic at the cellular level, many of which we suggest could be answered with a projectome, the detailed paths of every individual long-distance neurons with its downstream targets, relative to each other. 14. A projectome would at minimum classify potentially every single long-distance projection neuron in every brain region by its targets and the dendrite morphology. Moreover, a projectome would reveal every single long-distance pathway in a single brain, including some that have never been characterized before, the tilde operator strength of connection of every brain region on its target, and the numbers of projection neurons across different cortical and subcortical areas, essentially creating a Rosetta Stone for correlating with function or genetic expression in the future. By having a whole brain projectome, rather than a subdivision, e.g., visual cortex, will uniquely address variability in the organizations of brains by, for instance, quantifying how often the trajectory of individual neurons match the trajectories of nerve tracks, e.g., do individual axons branch when nerves branch and vice versa? How much variability is there in the composition of projection neuron classes and numbers of projection neurons across cortical and subcortical regions? Across animals, is variability in an individual projection to a particular brain region compensated by downstream pathways, i.e., if region X has a smaller projection to region Y in one animal, does the projection from region Y to Z increase to compensate? variability in disease states in response to the disease. We argue that such principles are hard, if not impossible, to reveal with either averaged or sparse sampling approaches. Finally, a hallmark of species with bigger brains, ranging from mice to humans, is variability in behavior and macroconnectivity, e.g., MRI, 15, 16, 17, 18. And thus we argue projectomes of large brains are well poised to address the fundamental question of how variability in long-distance communication between brain regions can underlie variability in behavior. Part of the reason such maps don't already exist is that there is no single imaging technique with the requisite resolution, sampling depth, and speed to image entire primate projectomes. Magnetic resonance imaging, MRI, approaches are fast, span whole brains, can be done in vivo, but lack resolutions to parse out long-range properties of individual neurons. Fluorescence approaches, e.g., light sheet 19, expansion 20, can resolve individual axons only if a small fraction, less than 5 to 10%, of axons are labeled, and in addition, fluorescence techniques often require genetic access for expressing fluorophores. Electron microscopy, EM, offers sufficient resolution to resolve individual myelinated axons, labels all neurons, and is species independent. But current 3D electron microscopy approaches are limited to volumes substantially smaller than whole primate brains, 21, 22. One promising option is to use synchrotron source X-ray imaging, which we have in the past demonstrated can image large volumes of brains at subcellular resolution, 23, and indeed can image entire mouse brains. Tilde operator 1cm3 at submicron resolution, 24. Synchrotron micro and nano ray computed tomography, XCT and XCT, have several advantages relative to standard optical microscopy and electron microscopy. Depth of penetration, 23. Infinite vertical tiling, 25. Speed of imaging tilde operator 10 MHz for XCT 23 and tilde operator 100 MHz for NXCT 26. Automated imaging, 25. Compatibly with post-hoc EM, 24. However, imaging an entire macaque brain, tilde operator 5 cubic centimeters, with X-rays poses at least two challenges. First, heavy metals used to stain brains for X-ray imaging, e.g., osmium, uranyl acetate, lead citrate, and epoxy resins need to diffuse through the entire brain. While it is now possible to stain entire mouse brains with osmium 27, 28, 29, the tilde operator 200-fold greater volume of macaque brains will still pose problems for chemical access primarily through diffusion. One possibility is transcardial perfusion of stains 30, but such approaches remain unreliable, hazardous, and thus unlikely to be used on scarce and valuable primate tissue. A second obstacle is that, even with the minimal scattering afforded by using X-rays, penetrating through multi-cm thick samples while maintaining focus and resolution remains problematic. A human projectome only exacerbates the problems by orders of magnitude. Thus, we developed a pipeline that entails subdividing brains with minimal loss, imaging each subvolume with synchrotron source X-rays, Figure 1, at resolution sufficient to reveal the trajectory of every MA and the morphology of every cell, and algorithms to trace MA in subvolumes and across cut surfaces. While the work presented here is a proof of concept rather than a whole projectome, it solves many critical hurdles first necessary to surpass and lays the path forward for following most, if not all, individual MAs over large volumes of a primate brain. Download figure. Open a new tab. Figure 1. Proposed pipeline for X-ray primate projectome mapping. Top left, 1. A brain is cut into pillars by first cutting the brain into large flat slices, e.g., A, B, C, D, etc., which are then cut a second time into thinner pillars, e.g., A1, A2, A3, A4, and B1, B2, B3, B4, etc. Top right, 2. Each pillar, after being stained with heavy metals and embedded in resin, is imaged by synchrotron source X-ray computed tomography, SXCT. Bottom left, 3 individual MAs, depicted as lines between each pillar, are traced within and across each pillar to reconstruct a full brain projectome. Bottom right, 4. After projectome mapping, the same pillars are compatible with large volume serial EM for focused connectomic reconstruction of synaptic circuitry. Middle, visual representation of primate brain subdivided into a series of pillars. 3 results. 3.1 Cutting brains with minimal loss. We first investigated whether a commercial system called the compressed-tome, a new microtome that compresses tissue as it's cut, developed for decreasing damage during cutting of living brain slices, 31, could be used to tracing individual MAs across tissue sections. The compressed-tome was recently used to track sparsely labeled fluorescent axons across serial macaque brain slices, 9, but because this approach relied on labeling sparsity to follow axons across cut surfaces, the surfaces of the cut sections were not evaluated, nor was the accuracy in following individual MAs across cut surfaces. We cut 300 m thick serial slices of cortex brain tissue and stained each slice with fluoromyelin, a fluorescent dye that labels individual MAs. 32, Figure 2a. We imaged opposing cut surfaces using confocal microscopy and found that we could easily identify many individual MAs across the cut surface, whether such axons were cut in cross-section, appearing as circles, or broadly parallel to the cut surface. Figure 2b, red box and green circle, respectively. We next compared the relative surface integrity of a standard vibrotome and compressed-tome brain section with scanning electron microscopy, SEM. Consistent with our ability to trace MA across cut surfaces, we found that the surface of compressed-tome sections were relatively preserved, with biological features like soma, nuclei, and individual MAs were reliably identifiable. Vibrotome section surfaces, however, appeared uniformly jagged without any discernible biological features. Supplementary Figure 1. Download figure. Open a new tab. Supplementary Figure 1. Compressed tome sections retain biological architecture on very surface of the cut section. A. Low-resolution SEM image of brain tissue cut with a vibrotome, left, and compressed tome, right. B. High-resolution SEM image of the surface of a vibrotome section, left, and compressed tome section, right. Note that in the compressed tome section, neuronal nuclei, dotted circle, and individual MAs, red arrows, can be readily visualized. Scale bar equals 1 mm, A 10 m, B. Download figure. Open a new tab. Figure 2. Individual MAs can be identified across brain sections cut with a compressed tome. A. Schematic of experimental design where a piece of tissue, A, is cut in half with a compressed tome to produce 2 sections, A1 and A2. MAs in sections A1 and A2 are fluorescently stained with fluoromyelin, and the matched surfaces are imaged, i.e., back of A1 and front of A2, by 2-photon confocal microscopy. B. Matched MAs from the back of A1 and the front of A2 can be manually identified, e.g., A1, 1 matches A2, 1 foot, etc. Red box and inset. Zoom of cluster 2 and 2 foot from A1 back and A2 front, respectively show 3 individual MAs that traverse the 2 sections. Green circle, an individual MA running in tilde operator plane with the cut surface. Dark circles are negative contrast from unstained soma. 3.2x CD tomography of large brain pillars. We next used the compressed dome to cut a 2x4 pillar array of macaque cortex with each pillar tilde operator 0.7 mm times 0.5 mm times 8 mm in dimension, spanning across primary and secondary visual cortices, V1, V2 respectively, and the white matter tracks between. Supplementary Figure 2. Each pillar was then stained and embedded in a hard plastic resin using established EM protocols, 33, which provide high contrast and physical stability for XCT imaging, 23, 24, 26, Figure 3a. We developed custom cylindrical molds that reduce the sharp edges of the resin to minimize X-ray scattering, 34, 35, 36, and that fit into a single field of view, FOV. Adjacent pillars were imaged using XCT at tilde 600 nm3 resolution, and acquisition took approximately 75 minutes to image 1.3 by 1010 voxels, i.e., 2.9 MHz. See methods for how to access full datasets. In both V1 and V2 gray matter, individual neurons and their primary dendrites are clearly visible, and in white matter, numerous individual MAs can be resolved, Figure 3b. Additionally, manual inspection showed large fiducials, e.g., blood vessels, that cross two of the pillars, allowing identification of adjacent cut surfaces. Figure 3c, Supplementary Movie 1. Download figure. Open a new tab. Supplementary Figure 2. Brain region excised for Primate Projectome. A coronal piece of primate brain was cut using a scalpel, and a small piece, red box, spanning V1 and V2 was further excised for compressed-tome sectioning. V1 and V2 was identified by locating the calcarine sulcus, dashed line. B. Left. Cartoon of tissue pillar array made from compressed-tome sectioning of V1/V2 brain tissue from A. Right. Bright field images of tissue pillar array spanning V1 and V2. Download figure. Open a new tab. Figure 3. Pillar for SXCT imaging. A shown is an individual pillar, 0.7 mm times 0.5 mm times 8 mm, spanning V1 and V2 with intervening white matter, green line, is prepared using the compressed-tome, stained for X-rays and embedded in a cylindrical resin mold prior to micron-scale X-ray imaging. B. Subcellular features can be readily identified, including dendrites, left and middle, red and green box, and individual MAs, right yellow box. Below 4x zoom-ins from each example from corresponding colored boxes can be seen throughout the volume. C. Adjoining surfaces of two opposing pillars, 1 and 2, are shown with a large blood vessel traversing across the gap. Scale bar equals 1 mm, A 10 m, B for mu-m-c. 3.3 tracing individual MAs across XCT pillars and estimating tissue loss. We next used the XCT datasets to ask whether individual MAs could be traced within and across the gap between two pillars, and to calculate the tissue loss between pillars. We traced 31 readily identified MAs that traverse the two pillars, Figure 4 and supplementary Figure 3. And we were able to follow 30 MAs across the entire 700 M plane of the two pillars, and the one that could not be fully traced demyelinated within the volume of Pillar 1, but could be fully traced through Pillar 2. The average length of MA traced was 1.4 millimeters, and in total we traced 86.99 millimeters of MA across the two pillars. We then used these reconstructed MAs to estimate the tissue loss, or kerf, between two pillars. Briefly, we fitted lines to each MA reconstruction that traverses the gap between the two pillars and estimated the distance between the ends of the MA reconstructions at the edges of each pillar. See methods. Using this method, we averaged the kerf over all 31 MAs as 697 nanometers, i.e., the loss from cutting is about 1 pixel of XCT data. Overall, we conclude that 1. Large pillars can be readily stained and automatically imaged by XCT. 2. There is minimal tissue loss between pillars. And 3. MAs can be manually followed throughout and across pillars, even with micron-scale X-ray imaging. Download figure. Open a new tab. Supplementary Figure 3. Montage of an individual MA traversing XCT pillars 1 and 2. Individual images that follow a MA, outlined in red, across the two pillars were stitched together to show the 2.2 millimeters path and ability to be traced entirely. Inset, zoom in showing a cluster of additional MAs, red asterisks. Roman numerals 1 to 4 correspond to zoomed in areas depicted below. Download figure. Open a new tab. Figure 4. Individual myelinated axons can be traced across XCT pillars. A. 2D image showing the entire length of pillars 1 and 2 overlaid with manual reconstructions of individual MAs that traverse the two datasets. Each colored line refers to an individual MA. B. Zoom in of A on region where individual MAs were traced across pillars 1 and 2. C. Zoom-ins of 3 different regions showing individual MAs traversing pillars 1 and 2. Red boxes. Regions further zoomed in and Schwann on right side with traversing MAs highlighted in red. Red asterisks mark blood vessels that traverse the two pillars. Scale bars equals 1.5 millimeters. A. 100 mc. 3.4 synchrotron nano X-ray tomography for tracing MA. Although XCD has the advantage of accommodating very large samples, even up to whole mouse brains, 24, we found the resolution of XCD was insufficient for automated segmentation of MAs and joining MA segments across the gap, both which would be necessary to scale this pipeline to whole brains. Thus, we next investigated nano X-ray tomography and NXCT imaging of brains as a higher resolution option for projectomics. We collected multiple NXCT datasets at multiple resolutions, using different nano X-ray imaging approaches and at different synchrotron beam lines. We ultimately created 2 datasets, a 30 nanometers resolution, tilde operator 50M3, volume from white matter in primates acquired on a transmission X-ray microscope, TXM. At the full-field X-ray imaging beamline of the National Synchrotron Light Source 2 at Brookhaven National Laboratory, BNL, and a 100 nanometers, tilde operator 250M, 3 volume of lower layers, L6, of mouse cortex at the European Synchrotron Radiation Facility, ESRF, using holotomography, 37, 26, 38. With 30 nanometers isotropic voxel resolution, we could reliably trace 100% of all MA within the volume in dense white matter and across demyelination events like nodes of Ranvier. We first asked how well such resolution could support tracing across kerfs. We produced computational gaps by removing slices from complete white matter NXCT stacks to mimic kerfs of different sizes, figure 5a, to determine how accurately humans could trace MAs across different sized divides, C methods. We removed 1.5 and 3M kerfs, tilde operator 2x, and 4x larger than the actual gap we calculated for the compressed tome sections, and had independent experts trace the same MAs across the 1.5 and 3M gap. Figure 5b, C. We found that experts agreed on the trajectories of 97% of the axons across a 1.5M gap, and that accuracy drops to 60% when the gap is 3M wide, 104 MA fragments, mean length tilde operator. 32M, total MA length, 3.3 millimeters. Although 30 nanometers NXCT provides superior resolution, such imaging is currently limited to volumes of tilde operator 50M3 and would necessitate cutting 10,000 S of approximate 1 millimeter diameter pillars to map even at 0.01% of macaque primate brain. Thus, we asked about mapping projectomes in a larger volume, but coarser resolution nanoholotomography X-ray dataset, 37, which we generated at ESRF at 100 nm 3 voxel resolution. We first verified that humans could reliably trace individual MA throughout the volume, 127 accurately axons traced, data not shown, C methods. Furthermore, we found that at 100 nanometers voxel resolution, dendritic arbors could be clearly traced, and demyelination events like nodes of Ranvier could be clearly visualized, figure 6a, supplementary figure 4. Download figure. Open a new tab. Supplementary figure 4. Nodes of Ranvier can be identified in NXCD. Zoom in of an individual myelinated axon, red dotted circle, with a node of Ranvier. Red arrow points to the node of Ranvier and green arrows point to the portion of myelin flanking the node of Ranvier. Scale bar equals 2.5 m. Download figure. Open a new tab. Figure 5. Individual MAs can be accurately traced across a computational curve up to 1 m. A. Schematic of experiment. The same 104 individual MAs were manually traced by 3 separate people in the same NXCD that is either 1 full, 2 minus 50 slices, 1.5 m gap, and 3 minus 100 images, 3 m gap. B. Manual reconstructions of the same set of MAs from the full NXCD dataset, top, or the NXCD dataset with a 1.5 m gap. Each color corresponds to an individual MA, and the matching colors from the full and 1.5 m curve are the same MAs across datasets. C. Plot of X. Computational curve distance, m, versus y. Accuracy of tracing across the computational curve. 97% of MA 1.5 m curve and 60% of MA can be traced across a curve of 3 m, mean length plus / STD 31.6 plus / dash 7.7 m. Total length traced equals 3.26 millimeters. Scale bars equals 5 m, A 10 m, B. Download figure. Open a new tab. Figure 6. 100 nm 3 NXCT has sufficient resolution for reconstruction of neuronal morphologies and individual myelinated axons. A 0.3D renderings from manual reconstruction of 3 neurons overlaid on a 3D plane view of the raw NXCT data. B. Single 2D image from the NXCT volume overlaid with segmentation of individual MAs. C. 3D rendering of segmented MAs from B. Scale bar equals 10, B. We next asked whether algorithms used for electron microscopy data could reliably segment MAs from 100 nm 3 NXCD data. We found that a state-of-the-art SOA EM segmentation algorithm, local shape descriptors, LSD 39, with minimal training could reliably identify nearly all MA profiles throughout a subvolume, 116 M3, of the NXCT dataset, Figure 6b. We manually inspected 100 random MA segmentations and found 81% of MAs were accurately segmented throughout the entire volume. Total length equals 3.3 millimeters. Average length equals 35 m. And all errors were splits, i.e., a single MA was labeled with 2 IDs. We found such splits were readily identified and fixed algorithmically. We first automatically identified putative split errors from the list of segmented objects by identifying tips of segments, i.e., where the segment ends. Second, we found MA tips that were within 250 nanometers and pointing at each other. We joined these ends, and subsequent visual inspection found that 100% of theses repair were correct. Finally, we asked how well can algorithms trace MA across curves of 100 nanometers. 3 NXCT data? To test this, we removed 19 sections, tilde operator 1.9 m, from the segmented volume, tilde operator 2.5, fold greater than the estimated real gap of 697 nanometers, removed all unique identifiers, and used matching algorithms and directional predictions described above to reconstruct MA across gaps. We found that our algorithm performed 94.4% optimally in matching MA segmentations across the 1.9 m divide, supplementary figure 5. While such computational gaps are likely idealized, i.e., they do not contain cutting distortions or other artifacts, they suggest that tracing the vast majority of MA across compressome-generated gaps is feasible. Thus, With minimal training, available algorithms for automatic segmentation appear sufficient for our proposed pipeline for projectome mapping of NXCD data. Overall, these datasets confirmed several points that suggest 100 nm3 data appears to be a sufficient resolution for projectome mapping of individual MAs and neuronal dendritic morphologies. Download figure. Open a new tab. Supplementary Figure 5. Individual MA segments can be joined across a computational gap. 100 nanometers resolution NXCT image showing the number of correct, green, incorrect, red matches, and those that didn't have a match across the gap, yellow, across a 1.9 m gap. Scale bar equals 10 m. For discussion. Here we outline an experimental pipeline for mapping many to all individual myelinated axons and cell morphologies across large brains using synchrotron source X-ray imaging. We addressed several challenges. Methods for sectioning brains with minimal tissue loss and preparing for X-ray tomography. Identifying the minimal resolution needed to trace all individual MAs within volumes and across tissue gaps. Automatic segmentation of individual MAs. Algorithms for joining segments of individual MAs across tissue gaps. We found that with current X-rays resolutions, i.e., 100 nanometers voxels, we can trace MA within pillars with near 100% accuracy and across pillars with 97% human accuracy and 94% algorithmic accuracy across gaps we estimate are twofold wider than the actual gap between compressed dome cut sections. Finally, we model the MA tracing success rate as P is almost equal to PCG times gap, where PCG is the fraction of axons within a pillar that are reconstructed without error. Gap is the fraction of axons successfully traced across a curved gap, and gap is the number of gaps jumped by the myelinated axon from end to end. Thus, the required success rate for an axon traversing on average 100 pillars or 10 centimeters for 1 millimeter wide pillars is PCG times gap greater than or equal to 97.8. Thus, we predict that algorithms could completely trace the long-distance MA projections of 10% of all MA, tens of thousands, in a CM3 volume, supplementary figure 6. This number could be increased with human intervention and error checking, which itself could be made more efficient by building expectations/priors for what MAs should look like using data collected from segmentation and error checking of MAs. These priors to identify possible sources of error, e.g., trifurcations, inappropriate loops, etc. Or to target human error checking could be scaled, using the rising number of online tools to error checking the algorithmic output of connectomic data, EYUwire, 40, Flywire, 41, and others 42, 43. Second, there is now a public challenge for auto-segmentation of MA through X-ray data, https://expressed grant-challenged.org, which will increase algorithmic efficiency. Download figure. Open a new tab. Supplementary figure 6. Estimated rates of successfully tracing myelinated axons across tissue pillars of different size. Plot of X, the number of neurons successfully traced versus Y, the total distance neurons are accurately traced for different rates of successful tracing across pillars. Estimates were performed for both tissue pillars that are either 1 millimeter, solid line, or 2 millimeters, dashed line, diameter. For 0.1 a projectome of a primate hemibrain. The projectome of an adult macaque hemibrain, conservatively estimated at tilde operator 5 by 3 by 5 centimeters, would create tilde operator 1500 pillars, 5 centimeters long and 1 millimeter wide, as described above. These pillars could be imaged at existing synchrotrons, like the Advanced Photon Source, APS, at Argonne National Lab, ANL, using XCT at 600 nanometers resolution in 78 days of continuous imaging. Tilde operator 50 MHz acquisition. Such data would allow for identifying every soma, tracing large fractions of dendritic arbors, tracing large caliber MA long distances, and detailing the full vasculature. A complete projectome, detailing the course of every MA in a volume, will likely require 100 nm 3-pixel size or better. We demonstrate such resolutions are currently achievable with NXCT, and some beam lines have achieved such resolutions over a millimeter of penetration, 38. One critical factor for future projectomes is that beam lines are often upgraded, sometimes increasing their brightness by an order of magnitude. For example, the APS at ANL is currently upgrading with an expected increase of 500x brightness. https://www.aps.anl.gov/aps-upgrade. With the upgrade, XCT at 600 nm3 of the hemibrain would take 16 hours and, assuming the major bottleneck in acquisition speed post-upgrade are detectors 44. And with the current availability of 6 GHz detectors, a 100 nanometers projectome would take 144 days. 5 methods. 5.1 Animal care and tissue processing. Perfusion procedures were followed according to animal regulations at the University of Chicago's Animal Resources Center, ARC, and approved IACUC protocols. Rhesus macaques were housed in accordance with the Guide for the Care and Use of Laboratory Animals, Public Health Service Policy, and the Animal Welfare Act and Regulations at the University of Chicago, a fully ALALIC-accredited institution. All procedures involving animal care and use were approved by the IACUC at the University of Chicago. 5.2 Fluoromyelin staining. Tissue sections were provided by Precisionary Instruments, makers of the compressed-tome. Paraformaldehyde fixed zebrafinch brain was cut into 200 m thick serial sections using the compressed-tome, keeping track of the order of sections. Tissue was then stained with fluoromyelin, Thermo Fisher Scientific, F34651, according to manufacturer's protocol. Stained sections were mounted with Prolong Gold anti-fade mountant, Thermo Fisher Scientific, P10144, and imaged with a Leica SP5 confocal microscope. 5.3 Tissue sectioning and staining. Primate brain tissue was prepared for EM as previously described, 33. Briefly, animals were deeply anesthetized until unresponsive. Primates were then transcardially perfused first with buffer, 0.1 m sodium cacodylate, pH 7.4, followed by fixative, 0.1 m sodium cacodylate, pH 7.4, 2% paraformaldehyde, and 2.5% glutaraldehyde. The brain was then extracted and post-fixed for 24 hours in fixative at 4C. A coronal piece of brain encompassing V1 was first cut out with a surgical scalpel, followed by further excision of an area spanning V1 and V2 for either vibratome or compressed-tome sectioning. The visual cortex was identified using the calcarine and lunate sulcus, and a region just ventral to the lunate sulcus was selected that spanned V1 and V2 with intervening white matter. Tissue was then stained with heavy metals, dehydrated and embedded in plastic for electron microscopy as previously described, 33. 5.4 Synchrotron X-ray computed tomography. 3 X-ray datasets were generated for the study. First, the XCT was produced at Argonne National Laboratory at Station 2 BM using 20 keV energy, 10x objective, and a detector size of 2448 by 2048. Each projection data volume was acquired at a 0.3s exposure per projection with a total of 3,000 angles, totally 15 minutes per volume. About 5 scans were required to span the 8 mm axis at 0.345 m pixel size, totaling about 75 minutes/pillar. Tomograms were reconstructed using publicly available scripts, https://github.com/tomography/tomopy-cli. Secondly, a NXCT data set was acquired with the Transmission X-ray Microscope, TXM, at the Full-Field X-ray Imaging Beam Line of National Synchrotron Light Source 2 at Brookhaven National Laboratory, 4546. The X-ray energy used in the experiment was set to 8.5 keV. The projection images of the sample were recorded at 600 angles in 180 with 0.3. Angle step. The exposure time for a single projection image was 0.1 s, and the time for a tomography scan was 1 minute. Tomographic images of the sample were reconstructed from the projection images with GradeRec reconstruction algorithm. 47. Implemented in a Python package TomoPy. 48. The reconstructed tomography images have isotropic voxel size of 20.6 nanometers. The third dataset was collected at the European Synchrotron Radiation Facility, ESRF, Grenoble, France, using holotomography and imaging parameters as previously cited, 37, 26, 38. 5.5 manual and automatic segmentation. MAs from XCT datasets were manually reconstructed in Gnosis, https://gnosis, APP, using skeleton tracings. 3D reconstructions of neurons from the 100 nanometers NXCT and MA tracing across a computational curve of the 30 nanometers NXCT data set were done in WebGnosos 49. https://webgnosis.org. Automatic segmentation of MA from the 100 nanometers NXCT was done using the previously published local shape descriptor algorithm, 39, which can be found here. https://github.com/funklab/LSD. 5.6 XCT gap estimation. To estimate the gap between the two adjacent pillars sectioned with a compressed-tome, 31 myelinated axons were identified that traversed the gap and skeleton reconstructed using Gnosis. Then we measured how strong the direction of the edge closest to the gap correlated with the edges as the skeleton tracing moved further away from the gap. To get a direction predictor of the skeleton as it enters the gap, we used the last 5 edges closest to the gap and calculated the weighted average direction according to the correlation coefficient. Next, we extended the skeleton across the hypothesized gap from both sides and varied the gap width to minimize the residual tension using the downhill simplex algorithm, 50. To measure the residual tension, we iterated over all axons, temporarily removed that axon and matched the axon stubs coming from pillar 1 onto the axon stubs coming from pillar 2 using the Klopftalker scheme 51. Because the relative alignment of the two pillars is not exactly known. We then used this interpolation to predict the position of the temporarily removed axon stub from pillar 1 and added the distance of its terminal to its partner's terminal from pillar 2 to the overall tension term. 5.7 Repairing MA segmentation splits. To repair splits, we use the skeletons from manual annotations in the XCT datasets that traverse pillars 1 and 2 as described in the methods section, XCT gap estimation. We identified all endings of skeletons and measured the pairwise distance between all endings of all skeletons. We matched endings by stepwise increasing distance up to a cutoff of 250 nanometers and at every step matching endings that had a smaller distance. We rejected tentative ending pairings if the dot product of the two normalized direction vectors was larger than SQRT, 0.5. We produced an NML file detailing all matchings created this way and used WebGnosis, 49, to manually assess the performance of this matching. 5.8 Connecting MAs across a computational gap. To estimate how effectively we can reconstruct axons over a gap, we used the 100 nm 3 NXCT data and manually removed 19 slices, 1.9 m, creating two subdatasets A and B. We then used the Kimimaro algorithm, https://xenodo.org/record/5539913 to skeletonize the segmentations derived automatically. Figure 6b. For each terminal on the surface of this artificial gap, we calculated the distance of each connected node along the skeleton using the Dijkstra shortest path algorithm 52, and measured average position of all connected nodes between 7 and 7.5 m away. If none were, we substituted it with the position of the most distant node. We used the connecting vector from this average to the surface node as a direction predictor and extended the skeletons tentatively from both sides halfway into the gap. If this connecting vector was longer than 5 times the gap width, it was rejected and a vector orthogonal to the cut surface was used instead. This led to 374 skeletons coming from the A side of the artificial gap and terminating halfway into the gap, and 383 skeletons coming from the B side of the artificial gap and terminating halfway into the gap. From here on, we considered these endings only in this halfway plane. We measured the pairwise distance of all endings from A and from B. We then used the Hungarian algorithm 53 to create a least cost matching for these potential pairs, using the square root of the distance as the cost. We counted the number of correctly matched pairs and calculated the performance metric as a percentage of the highest theoretically achievable score. 5.9 Data availability. All datasets are freely available for download at BossBD, https://bossdb.org. Acknowledgements. We would like to sincerely thank Jianchong for providing compressed home sections. This research used the full-field X-ray imaging beamline 18 ID of the National Synchrotron Light Source 2, a U.S. Department of Energy, DOE, Office of Science user facility operated for the DOE Office of Science by Brookhaven National Laboratory under contract No. DSC 00127004. References. 1. Michael J. Horowitz, Ed Esline, Angela Elgilizet Bongartz, Elaine H. Shin, Lydia Yang, Jeremy Ameller, Louis N. Vanda Lagemont, Kimberly A. Smith, Amanda Ebert, Zachary L. Riley, et al. An anatomically comprehensive atlas of the adult human brain transcriptome. Nature, 489. 7416 391, 399, 2012. Crossref PubMed Web of Science Google Scholar. 2. Kotranemans, Hartmut Mollberg, Sebastian Bludau, and Carl Zillis. Ulich Brain, a 3D probabilistic atlas of the human brain's cytoarchitecture. Science, 369, 6506 988, 992, 2020. Abstract/free full text Google Scholar. 3. Daniel J. Felleman and David C. Van Essen. Distributed hierarchical processing in the primate cerebral cortex. Cerebral Cortex, New York, New York. 1991, 1, 47, 1991. Crossref PubMed Web of Science Google Scholar. 4. Kim A. Meyer, Martijn D. Steenwijk, Lindedau, Menno M. Skunheim, and Jeroen J. Giegerts. Long-range connections are more severely damaged and relevant for cognition in multiple sclerosis. Brain 143, 1150, 160, 2020. Crossref PubMed Google Scholar. 5. Martijn van den Heuvel and Olaf Sporns. A cross-disorder connectome landscape of brain disconnectivity. Nature Reviews Neuroscience 20, 435, 446, 2019. Crossref PubMed Google Scholar. 6. Kuzing, Jianan Kong, Gao Xiaoyang, Jingqing Li, Juanxia Han, Yao Wang, Estate Emso Kadza, Manuel F. Casanova, and Xiao Li. Disrupted Brain Network in Children with Autism Spectrum Disorder. Scientific Reports 7, 116253, 2017. Google Scholar. 7. Yanivisov, Heidi Johansenberg, and Michelle Thibot Schotten. The role of diffusion MRI in neuroscience. NMR in biomedicine, 32, for E3762, 2019. Google Scholar. 8. Christine J. Charvet, Kwadwo Ofori, Christine Backham, Jianli Sun, Melinda S. Madril, Konhak Matyar, Brian Ledlow, and Andre J. van der Kauw. Tracing modification to cortical circuits in human and non-human primates from high-resolution tractography, transcription, and temporal dimensions. Journal of Neuroscience, 42, 1837-49, 3767, 2022. Abstract/free full text Google Scholar. 9. Fang Su, Yenshen, Lufin Ding, Chao Yuyang, Heng Tan, Hao Wang, Qingyuan Zhu, Rui Su, Feni Wu, Yanyang Xiao, et al. High-throughput mapping of a whole rhesus monkey brain at micrometer resolution. Nature Biotechnology, 39, 12-15-21, 15-28, 2021. PubMed Google Scholar. 10. Nicholas W. Plummer, Daniel J. Chandler, Jean M. Powell, Erica L. Scapini, Barry D. Waterhouse, and Patricia Jensen. An intersectional viral genetic method for fluorescent tracing of axon collaterals reveals details of noradrenergic locus coarealis structure. Enuro, 7, 3, 2020. Google Scholar. 11. Tamily A. Weissman, Joshua Arsenesh, Jeff W. Lichtman, and Jean Livet. Generating and imaging multicolor brainbow mice. Cold Spring Harbor Protocols, 2011, 7 PDB, Top 114, 2011. Google Scholar. 12. James H. Marshall, Takuma Mori, Christina J. Nielsen, and Edward M. Calloway. Targeting single neuronal networks for gene expression and cell labeling in vivo. Neuron, 67, 4562, 574, 2010. Crossref PubMed Web of Science Google Scholar. 13. Chase A. Weinholtz and Michael J. Castle. Intersectional targeting of defined neural circuits by adeno-associated virus vectors. Journal of Neuroscience Research, 99, 4981, 990, 2021. Crossref PubMed Google Scholar. 14. Narayan and Kasthuri and Jeff W. Lichtman. The Rise of the Projectome. Nature Methods, 4, 4307, 308, 2007. Google Scholar. 15. Sophia Mueller, Donhong Wang, Michael D. Fox, V.T. Thomas Yeo, Jorge Sepulcher, Mert Arsabanku, Rebecca Shafi, Jiel Liu, and Hexing Liu. Individual variability in functional connectivity architecture of the human brain. Neuron, 77, 3586, 595, 2013. Crossref PubMed Web of Science Google Scholar. 16. Eyal Bergman, Xenia Goffman, Alexander Kavashansky, and Itamar Khan. Individual variability in functional connectivity architecture of the mouse brain. Communications Biology, 3-1738, 2020. Google Scholar. 17. Liang Ma, Lixia Tian, Tianyu Hu, Tianzi Jiang, and Nanming Zhua. Development of individual variability in brain functional connectivity and capability across the adult lifespan. Cerebral cortex, 31, 83925, 3938, 2021. Google Scholar. 18. Mohammed El Sagir and Kathy J. Price. Interpreting and utilizing intersubject variability in brain function. Trends in Cognitive Sciences, 22, 6517, 530, 2018. Crossref Google Scholar. 19. Hiroki Rueda, Hans Ulrich. Pavel Austin, Michael Enikonomo, Jairam Chandrasekhar, and Philip J. Keller. whole brain profiling of cells and circuits in mammals by tissue clearing and light sheet microscopy. Neuron 106, 3369, 387, 2020. Crossref PubMed Google Scholar. 20. Emmanuel D. Karagiannis and Edward S. Boyden. Expansion Microscopy, Development and Neuroscience Applications. Current Opinion in Neurobiology, 50:56, 63, 2018. Crossref PubMed Google Scholar. 21. Jeff W. Lichtman, Hans-Peter Pfister, and Nir Shavit. The Big Data Challenges of Connectomics. Nature Neuroscience, 17, 11, 1448, 1454, 2014. Crossref PubMed Google Scholar. 22. Larry F. Abbott, Davi D. Bach, Edward M. Calloway, Winfried Denk, Catherine Duloc, Adrian L. Fairhall, Isla Feet, Kristen M. Harris, Moritz Helmstedter, Viren Jain, et al. The Mind of a Mouse. Cell 182, 6 1372, 1376, 2020. Google Scholar. 23. Eva L. Dier, William Gray Ronkel, Judy A. Prasad, Hugo L. Fernandez, Daga Gersoy, Vincent A. Andrade, Camel Fetza, Xianghui Xiao, Joshua T. Vogelstein, Chris Jacobson, et al. Quantifying mesoscale neuroanatomy using X-ray microtomography. In Neuro 4, 5, 2017. Google Scholar. 24. Sean Foxley, Vandana Sampath Kumar, Vincent A. Andrade, Scott Trinkle, Anastasia Sorokina, Katrina Norwood, Patrick Law Riviere, and Narayan and Kasthuri. Multimodal imaging of a single mouse brain over 5 orders of magnitude of resolution. Neuroimage 238:118250, 2021. Google Scholar. 25. Rafael Vescovi, Mingdu, Vid Andrade, William Scullin, Daga Gersoy, and Chris Jacobson. Tomasaic, Efficient Acquisition and Reconstruction of Teravoxel Tomography Data using Limited-Size Synchrotron X-ray Beams. Journal of Synchrotron Radiation, 25, 5 1478, 1489, 2018. Google Scholar. 26. Aaron T. Kwan, Jasper S. Phelps, Logan A. Thomas, Tri M. Nguyen, Julie Hahn, Chao Lin Chin, Anthony W. Azevedo, John C. Tuthill, Jan Funk, Peter Cloetens, et al. Dense neuronal reconstruction through X-ray holographic nanotomography. Nature Neuroscience, 23, 12, 1637, 1643, 2020. Crossref Google Scholar. 27. Sebastian Stroh, Eric W. Hammersmith, David W. Tank, H. Sebastian Sung, and Adrian Andreas Wanner. In situ X-ray assisted electron microscopy staining for large biological samples. Elife 11, E72147, 2022. Google Scholar. 28. Kuensong, Jur Weifeng, and Moritz Helmstedter. High contrast N-block staining of mouse whole brain and human brain samples for M-based connectomics. Nature Methods, pages 1 to 5, 2023. Google Scholar. 29. Sean McCullough, Jonas Binding, and Winfried Denk. Staining and embedding the whole mouse brain for electron microscopy. Nature Methods, 9, 121198, 1201, 2012. Google Scholar. 30. Sanford L. Pillay, Smjee Russell, Spencer Gordon Jr., and Mary A. Grillo. Fixation of neural tissues for electron microscopy by perfusion with solutions of osmium tetroxide. The Journal of Cell Biology, 12, 2385, 410, 1962. Abstract/free full text Google Scholar. 31. Hadia M. Abdilal, Hyan O. Kim, Reese Wagstaff, Ryoko Sawahara, Peter J. Southern, and Pamela J. Skinner. Comparison of vibratome and compressed-tome sectioning of fresh primate lymphoid and genital tissues for in situ MHC tetramer and immunofluorescence staining. Biological Procedures Online, 17:1, 8, 2015. Google Scholar, 32. Paula C. Monsma and Anthony Brown. Fluoromyelin red is a bright, photostable, and non-toxic fluorescent stain for live imaging of myelin. Journal of Neuroscience Methods, 209, 2344, 350, 2012. Cross-ref PubMed Web of Science Google Scholar, 33. Yunfen Hua, Philip Laserstein, and Moritz Helmstedter. Large volume en-bloc staining for electron microscopy-based connectomics. Nature Communications 6, 17923, 2015. Google Scholar, 34. Axel Lang, Andreas Kupsch, Bernd Muller, and Manfred P. Henschel. Edge Artifacts of Radiographic Images by X-ray Refraction, 2012. Google Scholar, 35. M. Strobel and Kardjalov, A. Hilger, Kuhn, G. Free, and I. Menke. High-resolution investigations of edge effects in neutron imaging. Nuclear Instruments and Methods in Physics Research Section A, Accelerators, Spectrometers, Detectors, and Associated Equipment, 604, 3640, 645, 2009. Google Scholar, 36. Samantha J. Borland, Julia Benson, Nick Ashton, Sheila E. Francis, Keith Brennan, Michael J. Sherratt, Philip J. Withers, and E. Canfield. X-ray microcomputed tomography, an emerging technology to analyze vascular calcification in animal models. International Journal of Molecular Sciences, 21, 1245-38, 2020. Google Scholar, 37. Anna Kumchenko, Christos Bikis, Alexandra Pakurianu, Simone E. Heber, Peter Thalmann, Hansdale, Gabriel Schweghauser, Jürgen Hensch, Stefan Frank, Magdalena Muller-Gerbel, et al. Hard X-ray nanoholotomography, large scale, label-free. 3D neuroimaging beyond optical limit. Advanced Science, 5, 617000694, 2018. Google Scholar, 38. Miriam Anderson, Hans Martin KJER, Jonathan Raphael Patino, Alexandra Pakurianu, Bent Packenberg, Jean-Philippe Tiran, Maurice Tito, Martin Beck, Anders Bjorholm Dahl, Vedrana Anderson Dahl. Et al. Axon morphology is modulated by the local environment and impacts the non-invasive investigation of its structure-function relationship. Proceedings of the National Academy of Sciences, 117, 5233649, 33659, 2020. Google Scholar, 39. Arlo Sheridan, Tri M. Nguyen, Dib, Wei-Chung Alan Lee, Stefan Salfeld, Srinivas C. Taraga, Yuri Manor, and Jan Funk. Local shape descriptors for neuron segmentation. Nature Methods, 20, 2295, 303, 2023. Google Scholar, 40. http://iwire.org. 41. https://flywire.ai. 42. Jonathan Zung, Ignacio Tardival, Kai-Su Klee, and H. Sebastian Zung. An error detection and correction framework for connectomics. Advances in Neural Information Processing Systems, 30, 2017. Google Scholar, 43. Daniel Hain, John Hoffer, Brian Matejek, Adi Suisapelig, Ali K. Alawami, Lee Kamensky, Felix Gonda, Egan Meng, William Zhang, Richard Schalek, et al. Scalable Interactive Visualization for Connectomics. In Informatics, Volume 4, page 29. MDPI 2017. Google Scholar. 44. Mingdu, Zichao Di, Daga Gersoy, R. Patrick Sien, Yevgenia Kozarovitsky, and Chris Jacobson. Upscaling X-ray nanoimaging to macroscopic specimens. Journal of Applied Crystallography, 54, 2386, for 01, 2021. Cross-ref Google Scholar. 45. Xianghui Xiao, Mingyuan Ge, Chongyin, Feng Wang, Li Jiezhou, Feichen, Yuchen Karenchin Wiegart, and Wakit Li. Full-field X-ray imaging, a workhorse microscopy beam line at NSLS2 for materials science researches. Microscopy and Microanalysis, 25, S2386, 387, 2019. Google Scholar. 46. Mingyuan Ge, David Scott Coburn, Yevgeny Nazaretsky, Weihai Su, Kazimierz Goffrin, Hui Juan Su, Xi Jianin, and Wakit Li. 1-minute nanotomography using hard X-ray full-field transmission microscope. Applied Physics Letters 113, 8, 2018. Google Scholar. 47. Betsy A. Dowd, Graham H. Campbell, Robert B. Marr, Vivek Vinagarkar, Samir Vidibnis, Lisa Axe, and D. Peter Siddons. Developments in synchrotron X-ray computed microtomography at the National Synchrotron Light Source. In Developments in X-ray Tomography 2, Volume 3772, pages 224 to 236. SPY, 1999. Google Scholar. 48. Daga Gersoy, Francesco da Carlo, Xianghui Xiao, and Chris Jacobson. Tomopy, a framework for the analysis of synchrotron tomographic data. Journal of Synchrotron Radiation, 21, 5/11/88, 1193, 2014. Cross-ref PubMed Google Scholar. 49. Kevin M. Boergens, Manuel Berning, Tom Bocklish, Dominic Bronlian, Florian Draich, Johannes Fraunhofen, Tom Harold, Philip Otto, Norman Rezepka, Thomas Workmeister, et al. Web Nasos. Efficient online 3D data annotation for connectomics. Nature Methods 14, 7691, 694, 2017. Google Scholar. 50. John A. Nelder and Roger Mead. A Simplex Method for Function Minimization. The Computer Journal, 7, 4308, 313, 1965. Cross-ref Web of Science Google Scholar. 51. R.W. Clough and J.L. Tucker. Finite Element Stiffness Matrices for Analysis of Plates in Bending. Proceedings of Conference on Matrix Methods in Structural Analysis, pages 515 to 545, 1966. Google Scholar. 52. Etzker W. Dijkstra. A Note on Two Problems in Connection with Graphs. In Etzker Viba Dijkstra, His Life, Work, and Legacy, pages 287 to 290. 2022. Google Scholar. 53. Harold W. Kuhn. The Hungarian Method for the Assignment Problem. Naval Research Logistics Quarterly, 2, 1 to 283, 97, 1955. Cross-ref Google Scholar. Back to top. Previous next. Posted October 24, 2023. Download PDF. Print/save options. Supplementary material. Email. Share. Citation tools. Audio abstract. Get QR code. Subject area. Neuroscience. Reviews and context. 0. Comment. 0. Trip peer reviews. 0. Community reviews. 1. Automated services. 0. Vlogs/media. 0. Author videos. Subject areas. All articles. Animal behavior and cognition. Biochemistry. bioengineering, bioinformatics, biophysics, cancer biology, cell biology, clinical trials asterisk, developmental biology, ecology, epidemiology asterisk, evolutionary biology, genetics, genomics, immunology, microbiology, molecular biology, neuroscience, paleontology, pathology, pharmacology and toxicology, physiology, plant biology, scientific communication and education, synthetic biology, systems biology, zoology. Asterisk, the clinical trials and epidemiology subject categories are now closed to new submissions following the completion of BioArXiv's clinical research pilot project and launch of the dedicated health sciences server MedRxiv. Submit medr14.org. New papers that report results of clinical trials must now be submitted to MedRxiv. Most new epidemiology papers also should be submitted to MedRxiv, but if a paper contains no health-related information, authors may choose to submit it to another BioArXiv subject category, e.g., genetics or microbiology. Reviews and context X. Comments 0. Trip 0. Community 0. Automated 1. Blogs/media 0. Video 0.